Hi everyone, and big thanks to the organizers for the kind invitation and for having me as part of this wonderful group of speakers. As mentioned, my name is Barbara Hollunder, and I recently completed my PhD in medical neurosciences in the network stimulation lab in the Department of Neurology at Charité Berlin under the supervision of Andreas Horn. And the focus of our research is on deep brain stimulation, which is an invasive neuromodulation technique that is best known as a treatment option for severe forms of neurological or psychiatric disorders. But as I hope to outline in my talk today, studying the effects of deep brain stimulation on the human connectome by means of neuroimaging may also open a unique window into which brain circuits can become dysfunctional in these conditions and how we can optimize their treatment. But before we dive right into the science, I'd like to do my flash talk now, so to speak, because we're such an interdisciplinary crowd. I thought it would be good to have some insight into where I come from and what my expertise is. By training, I'm a psychologist with a background in cognitive neuroscience, and I studied psychology at the University of Vienna, with some stopovers also in Nice, London, Berlin, and Dublin. And there, I also worked as a research assistant for the chair of cognitive psychology and at the clinical fMRI study group at the Medical University of Vienna. And this is also where my enthusiasm for brain stimulation and neurological disorders and neuroimaging first took shape. And after that, I then had the opportunity to join the structured PhD programs of the Einstein Center for Neurosciences and the Berlin School of Mind and Brain. And these amazing program support gave me the opportunity to choose a lab for my PhD that truly aligned with my research interests. And I was immediately fascinated by the outstanding research agenda and also the very welcoming atmosphere of the network stimulation lab, which is led by Andreas Horn. And what's unique about our lab is that it, at least for the last couple of years, has had both a Berlin and a Boston branch. The Berlin arm of the lab is part of the movement disorders and neuromodulation section within the Department of Neurology at Charité Berlin and led by Professor Andrea Kühn. And the Boston arm is embedded within the Center for Brain Circuit Therapeutics at Brigham and Women's Hospital and Harvard Medical School, which is spearheaded by Professor Michael Fox. And this dual setup, so to speak, has been quite a unique opportunity for me to already collaborate with the Boston Center over the course of my PhD, but I'm particularly excited about the prospect of joining Professor Fox's lab and the center for a postdoc quite soon. And then to complement my research interests also from the clinical side, I'm currently also training as a cognitive behavioral psychotherapist at the Center for Psychotherapy at the Humboldt University of Berlin. Of course, the science I'll share today is far from a solo effort, but instead it's the result of a close-knit collaboration with many dear colleagues from the Network Stimulation Lab, which you can see here on this photo, and also, of course, many international collaborators. And I'd like to give a big shout out to my colleagues, postdocs Lingfei Li, Garance Meyer, Nanditha Rajamani, and our mentor, Andy Horn, for their contributions to the work for my PhD I'll be sharing today. So the structure of my talk will be threefold, where in the first part, I hope to introduce you to the way we and others have started to explore invasive neuromodulation as sort of a beacon into dysfunction and its organization within the human connectome. And then second, I'd like to walk you through an example study of how we can apply these concepts and techniques to study dysfunction within 4 different brain disorders. And finally, I'd like to wrap up by discussing our lab's vision as to how we can build up on these findings to better personalize neuromodulation treatment. But now let's jump straight into the first chapter, which will focus on an introduction to the methods that we use in our lab and the concepts that we think about. So as mentioned, the broad theme here is neuromodulation, so ways in which we can actively engage with the brain to change its activity, so to speak. And neuromodulation involves both non-invasive techniques like transcranial magnetic stimulation, transcranial direct current stimulation, or focused ultrasound. But there's also invasive techniques like deep brain stimulation or targeted lesions. And what all of these techniques have in common is a causal element. So instead of just observing the brain, we're intervening and studying the measurable effects. But in this talk, I'd like to focus a bit more on the invasive side of the spectrum, specifically on deep brain stimulation. Now, here's a brief introduction to deep brain stimulation for those that may not be as familiar with the technique. Deep brain stimulation, or DBS in short, is an invasive technique that involves surgically implanting fine electrodes into subcortical brain areas, and these electrodes then remain there permanently and are powered through an implantable pulse generator, which you can imagine similarly to a pacemaker that is placed under the skin near the chest. And then through subcutaneous cables, the pacemaker delivers weak electrical pulses to the brain. These aren't felt by the patient, so they don't cause any pain, but they work quite effectively, if done right, to disrupt pathological brain activity in brain circuits to alleviate symptoms. So after the surgical electrode placement, multiple contacts along the electrodes can then be activated by a neurologist using an external programming device. And this then helps us to further fine-tune the stimulation and render the treatment more effective. However, with up to 16 contacts on newer electrode models, finding the perfect combinations of an activated contact and stimulation parameters represents a rather time-consuming challenge. But it also opens up a unique opportunity for neuroimaging-guided programming to render the process more efficient and personalized. So deep brain stimulation is perhaps most known for its applicability in movement disorders like Parkinson's disease, essential tremor, or dystonia. But over the last decades, it's also been repeatedly probed for a much broader spectrum of conditions. And these also involve quite severe forms of psychiatric disorders like obsessive-compulsive disorder or major depressive disorder. So it can be applied in cases where people have tried everything else from medication to psychotherapy, but without success. And of course, this comes with a lot of ethical responsibility. There's been a long and problematic history when it comes to psychosurgery, but today's methods and techniques are held to extremely high scientific and ethical standards. And when done right, DBS can be a real lifeline for people that have run out of options otherwise. I find it most intuitive to grasp these very dramatic effects that deep brain stimulation may sometimes have on patient outcomes on empirical examples like this Parkinson's disease patient. And this patient suffers from tremors. And on the left-hand side, you see his symptoms under the stimulation turned off, whereas on the right, the stimulation is turned on. And there's no dopaminergic medication given in either scenario, so they should be comparable. And I'm sure you will agree that the difference is quite striking. So the mere activation of the deep brain stimulation intervention leads to a drastic improvement in these alternating movements that are performed by the patient. However, and this is a major limitation, not every patient benefits from the intervention, or at least not to the same degree. And one important reason for this is that electrode placement can vary sometimes between patients. So specifically, the subcortical nuclei that we are targeting with deep brain stimulation in the brain are usually relatively small, so in the range of centimeters. And this means that even slight changes in these precise electrode positionings can lead to quite drastic changes in clinical outcomes. So here on the left-hand side, you see how the closer an active electrode contact is to a predefined optimal stimulation target, the better the clinical outcome would be across a cohort of patients. And now take patients 51 and 21. You see how their active contacts, highlighted in red, are only around 2 millimeters apart from one another. But one of them saw an improvement of over 80% in motor symptoms, whereas the other only benefited from lesser than 25% in improvements in motor symptoms, so to speak. So indeed, millimeters seem to matter a lot in deep brain stimulation. And this is not only the case for targeting, but also for the way in which we analyze these stimulation effects. And this means that we need to work with high quality imaging and also take into consideration individual differences in brain anatomy between patients. And we try to get there by relying on structural imaging for defining the individual anatomy. Structural MRI for the preoperative imaging, and then we use postoperative MRI or CT to reconstruct the electrode trajectories based on the artifact. We also integrate fine-grained brain atlases and anatomical expertise, as well as accurate image registration algorithms. And of course, like aligning multiple patients' brains to a common template space can be tricky, but it's an essential requirement if we want to meaningfully compare different electrode localizations across patients. And to help tackle challenges like these, our lab is developing an open source software that is called LeadDBS. And LeadDBS is specifically designed to allow researchers to accurately reconstruct these electrode trajectories based on the preprocessed patient-specific imaging, and then estimate stimulation volumes around the activated contacts, and finally compare them across patients on a group level. Over the last years, LeadDBS has steadily evolved, and now it also includes features for analyzing stimulation effects at different levels, from focused stimulation effects to volumetric whole brain networks and also fiber tracts. And by the way, don't let the DBS in the name fool you. LeadDBS might also provide quite some flexible and versatile tools for human brain mapping in general, as we think. So for example, it comprises a very comprehensive collection of subcortical atlases, structural and functional connectomes, flexible normalization algorithms, and also a very powerful 3D visualization environment. So this could be something to check out if this sounds interesting. And then once we've successfully pinpointed the precise electrode positions across patients and simulated the stimulation fields, the next step is to understand which brain circuits are being modulated by the stimulation and how they link to clinical outcomes. Conceptually, DBS is a relatively powerful tool when it comes to human brain mapping, because we are directly intervening in the brain, and this leads to changes in clinical changes in a clear temporal sequence. We can draw a clear causal inference or a relationship between a modulated circuit and a specific symptom. Now, DBS network effects can be studied from micro to macro scales, ranging from synapses to entire neuronal systems, but our lab typically focuses on the macro level, and we usually use neuroimaging as our primary readout. We've already heard a lot about that in prior talks, but neuroimaging-based connectivity measures are usually classified into both functional and structural connectivity. Functional connectivity measures on the one hand side usually rely on resting state fMRI, which measures temporal associations and brain activity between brain regions based on by detecting the blood oxygenation level, and then structural connectivity on the other side often relies on diffusion imaging, which measures the extent and direction of water diffusion in brain tissue to map white matter pathways. But for the sake of this presentation, I'll focus a bit more on structural connectivity. Now, analyzing the effects of DBS on streamlines at the group level is typically done by integrating a set of reconstructed DBS electrodes and their stimulation fields pooled across a cohort of patients with a 3D reconstruction of the human connectome and template space. And besides the diffusion imaging-based connectomes, and since we've heard yesterday that diffusion imaging can also be biased in terms of anatomical accuracy, we sometimes also rely on anatomical atlases, so tract atlases as the one shown here by Peterson and colleagues. And this is a really cool resource because it was created entirely by expert neuroanatomists who used a holographic VR setup to delineate these streamlines in anatomical space based on their expert knowledge, but also histological data. And then based on tract definitions like these, we can then apply group level statistics to each individual streamline separately. And to visualize this process in a bit more detail, I'd like to snip out one streamline out of this connectome to illustrate the process in a bit more detail, so to speak. So essentially, there's two scenarios that we can distinguish in this method that we apply here, which is called DBS fiber filtering, and which we often apply in our research. As you see here, the streamline is exclusively modulated by the electrodes of good responders, but less so by the ones of poor responders. So if we were to group the electrodes of the good responders, we see how they nicely align along the streamline, whereas the electrodes of the poor responders aren't even close to it, nor touching it. So this means that apparently modulating this particular streamline is quite important for improving our symptom, and we retain it for our model. And then in the alternative scenario, the relationship between tract modulation and symptom improvement is less clear. There's both a few electrodes of good but also poor responders that modulate the streamline, but there's also some others that don't touch it at all. And all of these observations taken together suggest that modulating this particular streamline is apparently less important for improving the symptom, and we can filter it out from the model. And this should also explain why we call this technique devious fiber filtering. Now, applying this strategy across all streamlines comprised within this connectomic resource eventually yields an optimal model of streamlines whose modulation is associated with the best treatment outcome across the cohort in this given symptom. And in other words, what this means is if we were to place an electrode into the brain and stimulate a set of streamlines, this should lead to maximal symptom improvement. So we can conceptualize it as an optimal symptom response tract. But now in this second chapter, I'd like to dive a bit deeper into how we can apply these concepts to study dysfunctional circuits on the example of 4 different brain disorders. So in this project, it all started out with a very fascinating conundrum, and this conundrum had been on my mind for quite some time. So in fact, the subthalamic nucleus, or SCN for short, is a very small subcortical nucleus that is only around 1 centimeter long, but it has become quite famous as an effective DBS target for especially movement disorders like Parkinson's disease or dystonia. And more recently, it's also becoming more and more studied to treat more severe forms of obsessive-compulsive disorder and Tourette syndrome. And just to give you an idea of how diverse these applications are, Parkinson's disease is predominantly marked by tremors and other motor difficulties like rigidity or postural instability, whereas dystonia involves uncontrollable muscle contractions. And then on the more neuropsychiatric side of things, OCD is most known for the presence of obsessive thoughts or repetitive actions that are known as compulsions. And Tourette syndrome involves these involuntary vocalizations or movements that are known as tics. So what exactly is it about this small nucleus that makes it so versatile as a treatment target? And how can we explain that we can stimulate it by means of deep brain stimulation to improve such a vast array of heterogeneous symptoms? And to me, this very much sounded like a paradox, and I found myself extremely intrigued to dive deeper and get to the bottom of that. And one hint that may already help us resolve this conundrum to some extent may come from the relatively widespread axonal input that the SCN receives from the entire frontal cortex. And as we know from preliminary insights in various disorders, DBS effects or the study of DBS effects might provide some insights into how exactly this function is neuroanatomically organized or implemented at the level of these frontal subcortical circuits. So with this idea in mind in this study here, we set out to explore whether DBS could help us effectively uncover dysfunctional neuroanatomy with therapeutic properties in these 4 disorders, so dystonia, Tourette syndrome, Parkinson's disease, and OCD. And again, the fact that electrodes are being placed in the same spatially very circumscribed target area for treatment of all 4 of these disorders allowed us to disentangle whether one and the same, or rather multiple different circuits, would be associated with the simulation. And more specifically, we approached the question at 2 levels. First, by relating the simulation impact to improvements in disease-specific cardinal dysfunction, which we did at the subthalamic voxel level, based on a technique termed DBS sweet spot mapping. And second, we applied the previously introduced technique of DBS fiber filtering to achieve models of optimal electrode connectedness for maximized symptom improvement per disorder category. Tackling this question was largely facilitated thanks to a big collaboration that spanned multiple surgical centers, and these generously shared invaluable invasive data of as many as 197 patients from 8 patient cohorts with us. And each of these patients had received bilateral implantation of electrodes targeting the SCN for treatment of either of these 4 disorders. Now, intriguingly, the mean electrode placement across most of these cohorts was already quite similar, namely within the posterior lateral portion of this nucleus. The only exception to this was formed by OCD, where surgeons are known to deliberately target a more anterior medial limbic associative domain of the nucleus. And this finding for us was this finding of at least relative similarity in the electrode positions was important for us to ensure that any differences in the dysfunctional conditions would not be merely driven by differentially placed electrodes, but rather reflected the actual importance of modulating specific sets of streamlines for improving specific types of outcomes. Now, when we map the stimulation effect at the subcortical level, so within the SCN, the sweet spot for dystonia expectedly fell within the posteriormost level of the nucleus, namely within the sensory motor zone, whereas the Tourette sweet spot was placed within the motor domain, the Parkinson sweet spot within the premotor territory, and finally also the sweet spot for OCD was in line with our expectations, namely it was located in the anterior medial limbic associative domain. And how about brain networks? Well, when only the plane connections of electrodes to the rest of the brain were considered, the resulting profiles that you can see here actually look quite similar. And this again was a nice confirmation of the idea that the average electrode placement across the cohorts was relatively similar at least. But then when we weighted these profiles by the relevance of their modulation for clinical improvement in these respective symptoms using DBS fiber filtering, a somewhat distinct pattern emerged. And interestingly, it further showed some level of specificity for each of the 4 cardinal dysfunctions. Now, putting these findings together across neuroanatomical levels, we can appreciate that the topographical organization of dysfunction within the subcortex, so at the level of the sweet spots in the SCN, was mirrored in a similar, but at the same time spatially inflated form at the level of the cortical projection sites of these somewhat segregated tracts. And we saw these cortical circuit mappings spanning a range of areas from sensory motor cortices in dystonia, motor and premotor areas in Tourette's, supplementary motor cortex in Parkinson's disease, all the way to the prefrontalmost areas in OCD. However, partial overlap in these circuit mappings also suggests that the dysfunctions in these disorders may not be entirely independent from one another. And now it might be precisely this spatial miniaturization effect from cortex to subcortex that could be the reason why we can modulate these large-scale brain networks using single small electrodes to treat these very different heterogeneous disorders if we place these electrodes at the site of so-called integrator hubs like the SDN. But again, this observation underlines the importance of meticulous targeting where millimeter differences make a critical difference. Now, based on our findings, it appears that we must be incredibly precise with our implantation choices, particularly in the subcortex, where different types of functional information are packed into tighter and more condensed anatomical spaces. By contrast, at the cortical level, these corresponding functional zones appear to be more spread out, which might at the same time also provide an exciting opportunity for non-invasive neuromodulation to tackle these dysfunctional circuits from a cortical angle. But how much, you might now wonder, would these findings actually extrapolate outside of the discovery cohort? Well, to address this, we were fortunate to pool an additional cohort of 32 Parkinson's disease patients that were generously shared with us by our collaborators from Würzburg. And something that is very important to stress out here is that these patients had been completely unseen by the initial tract model definition. So they had not been used to define the tract model. And indeed, if we use the overlap of stimulation volumes in these novel patients with the original tract model in Parkinson's disease to estimate clinical improvements in this entirely independent patient cohort, we get a good fit between estimates and clinical outcomes. So it seems that the more we modulate this tract model, the better the patient outcome will actually be. And then we applied a similar validation strategy also to the case of 35 additional OCD patients, and these had been pooled across 3 contributing centers. And also these patients had been entirely independent of the initial tract definition of this OCD tract model in this case. And again, higher overlap of these patients' stimulation volumes with the tract model was associated with an increased treatment benefit across the cohort. Now, seeing these, our tract models hold true also for unseen data, we began to wonder, could we potentially even enhance stimulation effectiveness by directly targeting these beneficial streamlines and using them as something like a roadmap for both surgical implantation, but also stimulation parameter optimization. And at this point, I'd like to share a little bit more about one of the 3 patient cases that were featured in our study for prospective validation of these models. And it highlights the story of Julia Hamm, who is a patient with an extremely severe form of OCD, who found renewed hope thanks to circuit-refined DBS reprogramming that was performed at MGH in Boston by Drs. Darian Doherty and Margaret Jensen and team. Now, Julia, for many years, had unfortunately been unable to eat and drink on her own. because of contamination fears, and that came to a point where she relied on a feeding tube, and she at some point also couldn't get out of a wheelchair. And despite multiple different treatments being probed, from medication to psychotherapy, nothing really helped, and her symptoms remained severe. So eventually DBS was probed as a last resort treatment, so to speak. But unfortunately, as you can see here, under these conventional clinical stimulation settings, Julia's benefit from the intervention had been rather minimal. And indeed, once we mapped our optimal streamline model to Julia's brain to understand how her electrode related to it, it turned out that her stimulation volume under these clinical settings did not encapsulate these beneficial streamlines. However, once we optimized these stimulation settings for her stimulation volume to fully encapsulate these beneficial tracts, and the collaborators in Boston implemented those on her stimulation device, her symptoms improved dramatically. And I'm quite happy to share that I recently had the wonderful news that Julia apparently continues to do extremely well under these new settings. But of course, while this is very encouraging to hear, these initial findings are anecdotal in nature and should be seen rather as initial stepping stones towards more large-scale and systematic prospective clinical validation trials. Amazingly, Julia is also actively raising awareness for her disorder and for this treatment option. And she even agreed for her story to be shared on TV, and she also gave an interview. So if this sounds interesting to you, then I'd love to invite you to check those out. But while these initial clinical potentials were already really exciting to see, we believe that the story doesn't end there quite yet. So in this third and final chapter, I'd love to dive a bit deeper into our lab's vision for how we can build up on these initial mappings to bring them a bit closer to optimized personalization and neuromodulation. So one example among the mappings that I shared before that has been more extensively explored is this OCD response tract. After its initial characterization, it has been repeatedly validated across different surgical centers and also patient cohorts in different countries. And something that was quite interesting to see among these validations was that despite initially defined based on data of patients with OCD, work by Kara Johnson could show that this tract was also able to explain outcome variants in comorbid obsessive-compulsive behavior in patients with Tourette syndrome, so in a different disorder. So in a transdiagnostic sense, the therapeutic properties of stimulating these sets of streamlines might indeed cut across distinct disease categories. But beyond these co-occurring symptoms across disorders, different patients that are comprised or grouped into one and the same disorder category can often present with quite unique arrays of symptoms and comorbidities. So for instance, we know that OCD isn't just about obsessions and compulsions, but instead it's a very complex and heterogeneous disorder that also involves symptoms like anxiety, depression, or other comorbidities. And each of these symptoms may yet again map to distinct symptom network topographies. So that's exactly what we did in a follow-up project. We believed that we could zoom more closely into each of these tracts at higher levels of granularity to uncover even more targeted therapeutic mappings. We see this concept again here visualized on the example of OCD. So in this follow-up project, we set out to explore whether DBS could help us identify these differential mappings for different OCD subsymptoms, so to speak. And again, we were quite fortunate to pool a relatively large sample across contributing centers. This comprised many patients that had been targeted to very different sites. So in total, there were 6 different stereotactic targets that these patients had been operated on. But still, a very similar optimal global OCD response tract emerged. And indeed, it appears to not just be one uniform pathway, but instead it can be broken down further into a spectrum of symptom-specific bundles that are each linked to more granular symptom improvements. And we believe that a similar PRISM concept could also be applied to the other 3 disorders to study them in greater detail and also potentially in somatotopic fashion in the case of dystonia. And this is what my colleague Nandita Rajamani did also for Parkinson's disease. So to zero in more closely onto this global Parkinson's disease motor tract, she studied a relatively large cohort of Parkinson's disease patients who had been pooled across multiple surgical centers and who had received stimulation to the subthalamic nucleus target. And indeed, she found a gradient of these differential mappings for different Parkinson's motor subsymptoms. For example, improvements in tremor were associated with stimulation of tracts that mapped or connected to primary motor cortices and cerebellum, whereas bradykinesia and rigidity improvements were rather associated with stimulation of tracts connecting to supplementary motor and motor cortices and premotor cortices, respectively. So looking ahead, we believe that expanding this approach to encompass an even broader range of brain circuit disorders and at higher anatomical detail might allow us to create something like a target library or roadmap linking specific symptoms to distinct brain circuits. And this then might eventually help us delineate what we have tentatively started to term the human dysfunctome, in analogy to the genome and connectome concepts. And this is supposed to comprise the entirety of connections that can become dysfunctional in an array of different brain circuit disorders. And this again might open up an incredible wealth of possibilities for more personalized brain circuit therapeutics, not only in deep brain stimulation, but also in other neuromodulation modalities. But which steps are required to bring this concept into clinical settings? Well, our vision is that when a DBS patient first comes to the clinic, they undergo thorough symptom profiling. And from there, the relevant symptom network target templates could then be pulled from the library. And we could, at this stage, also apply a weighting step to account for the empirical intensities at baseline, but also for the subjective priorities to account for the fact or for specific symptoms being maybe more cumbersome for that respective patient. And finally, we could recombine these targets, these weighted targets, into one optimal simulation target by means of an algorithm. And to put this idea into action, my colleague Nandita Rajamani developed this really cool algorithm called ClearTune. Now, ClearTune takes the individual symptom profile of a patient and tries to figure out which DBS settings are most likely to address these best. And what you see in this video is how the algorithm sort of iterates through these recombinations of settings and active contexts, simulates stimulation fields that are corresponding to these settings, and also the tracts that are associated with them, and finally narrows down the options to one optimal configuration that is most that received the highest received the highest estimates for clinical benefit in that given patient with that specific configuration of symptoms. And this is just another example of how the algorithm tries to optimize for specific symptoms, in this case, bradykinesia, rigidity, tremor, and axial symptoms. And you will soon see how, after probing and jumping around between different options, the algorithm eventually narrows down its options and sticks with one specific stimulation volume that best addresses this specific subtype of motor symptoms. And this is just a small outlook. We believe that this circuit-based framework could also potentially help us harmonize treatment strategies across different neuromodulation modalities, both from the invasive but also non-invasive world. And this could be done based on shared circuit mechanisms. So, for example, if we were to identify one network that is particularly effective in treating a specific symptom by means of deep brain stimulation, we could also apply a probe it by means of non-invasive neuromodulation and see whether we can access it and access treatment benefit by stimulating it from a cortical angle. So we're nearing the end of my talk, but if there's just one metaphor that I'd love for you to take away from this presentation, it should be this. So I hope that these examples that I've just shared may have convinced you that we can indeed use connectomic deep brain stimulation sort of as a guiding beacon that could shine light from the depths of the brain to illuminate the organization of dysfunction within the entire human connectome at all levels between cortex and subcortex, and also at different degrees of granularity. And besides opening up a window into the human brain, we believe that these findings may also be of high clinical relevance for guiding us towards an improved and more personalized patient treatment. Now, if this got you curious, my PI is also hosting a really cool podcast called Stimulating Brains, where he chats with inspiring minds from the neuromodulation field. And concurrently, we're also running a monthly recurring virtual talk series that brings together the topics of connectomics and neuromodulation, but also gives a glimpse of the person behind the science. And we are also hosting not only senior speakers, so more established researchers from the field, but also give a stage to more junior researchers. So if this sounds interesting to you and you'd like to learn more, then we'd be very happy to have you join one of the upcoming sessions. And with that, I'd like to thank everyone very much for following along. I'm sure you'll agree with me that science takes a village. And as you can tell from the long list of collaborators and lab mates on the slide here, I was quite fortunate to have a relatively large one behind me for my PhD. Again, a particularly big shout out to my awesome colleagues from the Network Stimulation Lab, whose work this presentation was majorly built around. Ning-Fei Li, Garance Meyer, Nalitha Rajamani, and of course, our wonderful PI, Andy Horn. Last but not least, my deep gratitude is, of course, also owed to the patients for the study participation and to the many wonderful collaborators for generously sharing the data and expertise. Thanks again for having me, and if there's any question, I'm happy to have them.